Okay, welcome to video 8 and our introduction part of our course G08 on metagenomics in terms of viruses. And here we're going to look briefly at some aspects of basic functional analyses. So there's a wide variety of databases already outlined that from which we can derive functional information. And that is also introduced into the estimate the relative abundances of the different species. The classic ones are looking up information in KEGG and doing Go analysis or using BLAST to explore these things. For um, certain organisms, we could also be interested in horizontal gene transfer, although that's very, very rare in viruses, even in longer double-stranded DNA viruses. One thing that we will try in our practical is this tool called Logan, which has very, very fast alignment of short query of interest with most of the NCBI SRA. So this covers about 96% of the SRA based on the last publication instance. This spans about 50 petabytes of data and includes 384 terabytes of compressed context against which you can search using cloud-based infrastructure. You can search contigs and unitigs. And unitigs are variations seen at least twice in the reads. So the contigs are more defined set and unitigs are a broader set. You see that here the rate of increase in the SRA is very, very large, and same with the number of accessions, and thus having scaling down the amount of context that makes the search possible is very important. So what we're going to do is go to the Logan search website. We're going to search a FASTA query, though it can't take a sequence from a FASTA library. It uses a camera length of 31 for the search, and you can adjust the stringency threshold to restrict how many cameras are needed in a sample to have a match. So that means you can basically take your query of interest and get more information on that and the different matches. And this spans right across the tree of life. And what you can see in this diagram here with the circle is that for different organisms, although NCBI has a huge amount, the Logan database has somewhat more, or at least replicates what is present. And you see that the metagenomes come from very, very diverse areas. Obviously, there's biases in terms of sampling across human wastewater, human feces, and mice as well. You certainly see that it is a very broad representation of what we know already. Another one which we won't try, but you should be aware of, is the Big Fantastic Viral Database, which contains structural information for many, many viral species and their known fragments of genes or proteins. And this is important because AlphaFold doesn't include viruses. So viruses, there's no viral information there. And this predicted 351,000 protein structures with CloudFold, 62% are which are new. And therefore, you can use aspects of this information to inform your research. And what you can see here is the kind of breakdown of the different structures that are present on this database. It doesn't cover everything, but it covers a good spectrum. And a final point is the nuances of looking at comparing across different studies, which is complex, and there's a good paper on this, denoting the differences between looking at the relative abundances of different microbiomes across different studies, where the extent to which different microbes are sampled in a study is very, very crucial. And thus, if you're trying to aggregate data across different metagenomic information across different metagenomic studies, where the methods may differ, or indeed any different, or even if they're the same methods but have been done 2 years later, it's very, very difficult to make comparisons that are robust. And thus caution needs to be an important feature of any kind of investigation across different metagenomic studies. So that concludes our videos. Please see the lab-based videos for the more hands-on aspect.